Hi, I'm Caroline Clark, conference producer at Technology Networks, and I'm here to introduce our next talk, Complementary Gene Editing Approaches to Investigate Rare Mitochondrial Disease. I'm pleased to welcome Peter Kropp as your presenter. Peter earned his PhD in molecular physiology and biophysics at Vanderbilt University, with a focus on the gene regulatory networks governing pancreas development. He followed this with a postdoctoral fellowship at the National Institutes of Health in the National Institute of Diabetes and Digestive and Kidney Diseases. His postdoctoral work was focused on modeling rare mitochondrial diseases in the neonate C. elegans. Peter recently transitioned to a tenure-track position at Kenyon College, where he continues to expand on his postdoctoral work with C. elegans and mitochondrial dysfunction. A warm welcome to you, Peter. Following the presentation, we'll have a live Q&A session, and we'll welcome any questions that you may have. You can ask questions on the right-hand side of the video player, so please do go ahead and ask. You can also use this feature to connect with attendees and discuss the talk, so feel free to take advantage of the chat system by introducing yourself and sharing your thoughts during Peter's presentation. If you experience any technical difficulties during the presentation, please request help using the technical help icon to the left-hand side of the player. Now, without further ado, I will hand over to Peter to begin his presentation.
Well, thank you so very much, Caroline, and thank you to everybody at Technology Networks for this great invitation and this great meeting. And I'm very excited to tell you about the work that I started as a postdoc and is now ongoing in my fledgling research lab. So I study mitochondria and things that can go wrong with mitochondria. And if you ask anybody who has ever taken high school biology, they'll be able to tell you that mitochondria are the powerhouse of the cell. But there's a lot more to mitochondria than just the fact that they make ATP, although that is obviously a very important part of it. So I really like to ask these questions about what can go wrong and what that's going to do to the mitochondria and then the cells in which those mitochondria are resident. So this is important because of the fact that mitochondrial dysfunction can affect cellular identity as well as the function of that cell, that the responses to these, or this can generate stress, and the responses to that stress can be multicellular. And as somebody who's also a little bit trained in developmental biology, multicellular responses are very interesting to me. And the fact that mitochondria are extremely well conserved means that we can study them in these microscopic organisms and learn a lot about what could be happening in humans. So as was mentioned, my focus is on mitochondrial diseases. There are over 45 different disease families or mitochondrial disease families that have been identified to date, with anywhere from 2 to 10 individual diseases within that family. And worldwide, the incidence of these mitochondrial diseases is about 1 in 5,000 individuals. If we define that a little bit more narrowly, we can look at rare mitochondrial diseases. And in the United States, a rare disease is defined as one that has fewer than 200,000 patients. And you can see that these numbers don't actually change dramatically. We still have about 45 disease families, and the incidence is now about 1 in 5,500 individuals. So most mitochondrial diseases are in fact rare diseases. In my work, I focus on 2 of these disease families, one of which is iron sulfur cluster diseases, and the other are diseases of this poorly understood pathway that is mitochondrial fatty acid synthesis. I'm going to focus on one of the iron sulfur cluster diseases today, and this is called multiple mitochondrial dysfunction syndrome 1, or MMDS1. There's only 38 patients that have been identified with MMDS1. But what I think is important to remember is the fact that anything we learn about these individual rare diseases is not only going to be applicable to other diseases within the direct family, but also very likely to the diseases that fall under the greater umbrella of mitochondrial diseases. So I am hopeful and optimistic about the fact that everything that we learn is going to be able to be translatable beyond just the work that I do. To front load my talk with the central model, Again, focusing on MMDS1, which I'll give a little bit more information about in a second, but it's caused by a factor called NFU1, which handles iron sulfur clusters, these little yellow and red structures here. And it functions as a dimer to deliver those iron sulfur clusters to iron sulfur proteins, or ISPs. Those iron sulfur proteins will go and perform generally catalysis, so many of them are enzymes, either within mitochondria or within the cytosol. There's even a handful in the nucleus. But obviously, they need to receive these iron sulfur clusters in order to do their work. And in my work, what I've done is focused on a few specific mutants. These are the specific amino acid changes in NFU1 here, but they bind to these iron sulfur clusters differently, and that still leads to iron sulfur protein dysfunction, but via different mechanisms. Still, some of the phenotypes are going to be similar, including increases in reactive oxygen species, so oxidative stress, decreases in aerobic metabolism, and then, as I'll focus on in the back half of my talk, how this impacts neuromuscular dysfunction. So a little bit more information about multiple mitochondrial dysfunction syndrome 1, or MMDS1, said that this is caused by mutations to NFU1. This is the human version of the protein. When we're talking about the C. elegans version, you just have the hyphen between the letters and the number. As I was saying on the previous slide, mutations to NFU1 are going to impair iron sulfur cluster, or ISC, delivery. And this is an autosomal recessive disease, meaning that you need two mutated alleles in order for the disease to develop. But what's complicated is that 20 different mutations have been documented in NFU1. And the pathogenicity of these individual mutations is not very clear because about half of those patients are compound heterozygous, meaning that both alleles of NFU1 are indeed mutated, but it is different mutations in each allele. So that really complicates the understanding of just what could be happening in any given mutation to NFU1. MMDS1 is a rather tragic and severe disease. It is almost exclusively childhood lethal. It is neurodegenerative. So many of the individuals with MMDS1 initially develop normally, but starting around 6 months of age, start to develop symptoms of neurodegeneration, including the loss of motor control, so dystonia. There are also severe aerobic metabolism defects indicating that the mitochondria are indeed dysfunctional. So again, in this very simplified model, it's just again illustrating that NFU1 is delivering these iron sulfur clusters to the iron sulfur proteins. And I emphasize that because it is the dysfunction of these iron sulfur proteins that we actually see as phenotypes in my model, and not the direct effect of NFU1 itself. The way that I've chosen to study MMDS1 and NFU1 is by looking at their function in a small microscopic nematode called C. elegans, or C. elegans, or as I'll refer to them throughout most of the rest of the talk, just worms. You can see one of these worms crawling across the slide right over here. And worms are actually a fantastic model for understanding especially rare diseases, because about 80% of rare disease-causing genes are very well conserved between humans and worms. And they have an isogenic background, the worms that is, meaning that they are genetically identical other than whatever mutations we might introduce. Their cell biology is extremely well conserved. They are multicellular, which gives them an advantage over yeast. Sorry to my yeast friends. They are transparent, which allows us to do a lot of cool imaging. So we can see through this intact adult worm here, as well as we can see some embryos dividing in fluorescence imaging below. And what I'm going to focus on are some of the unique and extensive genetic tools that we have available, including RNA interference as well as CRISPR, both of which are extremely tractable in worms. And altogether, this makes them a great system for doing genetic and chemical manipulation. I'm going to show quite a few different genetic manipulations that I have done in my work. So when I first launched into this project, I started by trying to understand some of the differences between the different mutations that have been documented in MMDS1. So I used CRISPR to make those patient-specific mutations in worm NFU1 in the orthologous residues, all of which are perfectly conserved. I focused on 5 different residues, all of which are in the iron-sulfur cluster interaction domain. So this is showing part of the NFU1 protein. I'll remind you, it normally functions as a dimer, so there would be another subunit over here. Here we have our iron-sulfur cluster. But here are all of the mutations that I have modeled, many of which are impacting the charge or the polarity of the residues that are in this domain. And given that iron-sulfur clusters themselves are charged, this is not surprising that they would have negative effects. In these mutants, some of the gross phenotypes that were immediately obvious is that they experience larval arrest. So worms have a very stereotyped larval development. And like many mitochondrial mutants in worms, they arrest as larvae. They are sterile. So worms are also hermaphroditic. And the NFU1 mutants do not produce progeny on their own. This is again characteristic of mitochondrial mutants, as well as they have decreased lifespan. So it was immediately obvious to me that there was something going wrong here. But I wanted to get a little bit deeper into just what might be happening and to see if there was an allelic series at all amongst the 5 different mutants that I made. So I focused on what is the canonical way of looking at iron-sulfur cluster dysfunction, and that's to focus on one of the targets, which is a protein called the lipoic acid synthase, or LIAS. And it, in fact, needs not 1, but 2 iron-sulfur clusters that are delivered to it by NFU1. It makes this little adduct called lipoic acid, which it adds to 5 other enzymes, all of which are important for cellular metabolism. And the ones that get the most attention are the pyruvate dehydrogenase and the alpha-ketoglutarate dehydrogenase, which in worms are called DLST1 and DLAT1. We can look at this by performing a Western blot, which I'm showing here. And even though all 5 of these proteins get lipoic acid, if you use an antibody against lipoic acid, you normally only get 2 bands, and that's the pyruvate dehydrogenase and the alpha-ketoglutarate dehydrogenase. So if you look in the wild type sample here, you can see the strong lower band, which is DLST1, and a faint upper band, which is DLAT1. And that's what I would expect to see normally. And it should be immediately obvious that amongst these different mutants, there are differing degrees of lipoic acid dysfunction, or impairments in biosynthesis, I should say. And what was really weird and surprising was that in the mutants that are shown here on the right half of the image, including a full NFU1 deletion, that it wasn't just that this doublet disappeared, we actually had the appearance of this lower band, which I wasn't able to completely biochemically determine, but is the correct size for the lipoic acid synthase. So it seems like enough of the lipoic acid is getting made that the antibody can recognize it, but it is not getting completely made. It actually needs the sulfurs from these iron-sulfur clusters. So it's not getting completely made, and it's not then getting added to the different target proteins that need that lipoic acid. From here on out, I'm going to focus on 2 of these mutants. And this is the Gly147Rg mutant, so a glycine to an arginine, and Gly166cis, a glycine to a cysteine mutant. And I chose to focus on these for a couple of reasons. One is that these represent some of the most common of the mutations in MMDS1, with the Gly166cis mutant being the most common overall. The Gly147Rg is one of the other more common mutants, but I also chose to focus on these because both of them are hypomorphic. You'll notice that there's at least some lipoic acid that is getting delivered to those target proteins. And I thought that that was something that would be really interesting to look at, so that's why I have focused on those. The primary questions that I've been asked in this project, and I'm going to use as a framework for this talk, are how and why are the mitochondrial dysfunctional in these mutants, and do any non-metabolic factors contribute, meaning is it just ATP or not, as well as how does this mitochondrial dysfunction impact physiology? So with this being mitochondria, I did first look at their respiratory function, meaning the ability to undergo aerobic respiration. And I did this using what's called a seahorse bioanalyzer, where you put intact worms into a plate and it can measure the rate of oxygen consumption. That's the OCR on the y-axis here, which is going to be a good proxy of the rate of electron transport chain function and thus aerobic respiration. And what you'll notice is that the Gly147Rh mutant is perfectly normal, whereas the Gly166 cis mutant is severely impaired. So this was surprising to me, given the fact that there was some lipoic acid dysfunction in this Gly147Rh mutant, but clearly there was enough still being made that we could, or that they are capable of performing aerobic respiration. And as I said, this was done on intact worms, and I wanted to look a little bit more deeply. So to do that, I used a fluorescence imaging approach where I crossed in an allele that expresses the green fluorescent protein, or GFP, in all mitochondria in all of the cells of the worm, and then stained them with a dye called TMRE that only accumulates in functional mitochondria. This is more specifically a readout of the mitochondrial membrane potential, and mitochondria need to be hyperpolarized in order for that membrane to be functional, or for them to be functional, excuse me. And what you'll notice is that whereas we have this nice orange staining in the wild type animals, that it is diminished in the Gly147Rh mutant, and then almost completely absent in the Gly166 mutant, and that data is quantified over here. So this was really interesting to me because there was a disconnect between this TMRE staining of the Gly147Rh mutant and its respiratory function. Normally, it is, in fact, the electron transport chain that's responsible for maintaining that mitochondrial membrane potential, but it can be affected by other stressors. So it told me that there must be some other form of stress in this mutant that isn't necessarily present in the Gly166 cis mutant. And what I thought that might be is oxidative stress, that the generation of reactive oxygen species can affect this mitochondrial membrane potential. So I looked at this, the generation of reactive oxygen species, via what I have been calling an exogenous or ex vivo assay, where I ground up the worms, put them in a 96 well plate, and exposed them to this dye called H2DCFDA that in the presence of reactive oxygen species will fluoresce. When I generated this data, and this was over a time course, so we have time on the x-axis and we have relative fluorescence units on the y-axis, I indeed found that the Gly147Rh mutant had significantly more reactive oxygen species than wild type, or even the Gly166 cis mutant. And just so that this was a little bit easier to interpret, I only left one of the error bars on there, but this is indeed statistically significant. So it told me that yes, the reactive oxygen species must be at least a significant contributing factor to that loss of mitochondrial membrane potential. But why would it be that that mutant specifically is having this sort of effect? So I was trying to ask this question of what accounts for the allelic differences in Ross generation. And to try to understand it, I turned to the literature, where there's a group that is now my neighbor in Columbus, Ohio here, but at the time was halfway across the country. And they study NFU1 and also looked at some of these specific mutations and found that different NFU1 mutations observed in MMDS1 are capable of causing differences in dimerization of the NFU1 protein around the iron sulfur clusters. There was also the fact that oxygen, dioxygen, in mitochondria might be capable of reacting with iron sulfur clusters or labile iron, meaning just free iron within the cell, to generate reactive oxygen species. So I thought that perhaps these individual mutations were causing differences in handling of iron sulfur clusters, and that might result in the generation of the reactive oxygen species and other forms of stress. So to model that with these here, the Cowan group at The Ohio State University found that the ortholog of the Gly147Rh mutant does not dimerize as effectively. and that this could expose bound iron sulfur clusters to either dioxygen or other oxidant factors, including hydrogen peroxide, to generate reactive oxygen species. The Gly166 cis mutant, this extra cysteine actually causes there to become a disulfide bond across the dimers, so that it binds more tightly. It cannot release the iron sulfur clusters. And that is then not necessarily going to be generating these ROS on its own, but both of these are going to impair iron sulfur cluster delivery. The other thing that could be happening, and I'm not showing the data today, it was in a paper I published a couple of years ago, but that I was very interested in this idea of, well, if iron sulfur clusters are dysregulated, maybe this is increasing the pool of labile iron inside of these cells. And that iron itself can then also react to generate reactive oxygen species via something called the Fenton reaction. And I did find that these mutants do have elevated iron in an elevated iron response, likely due to the fact that they're not delivering these iron sulfur clusters properly. But collectively, these iron sulfur proteins are dysfunctional. This is leading to the development of oxidative stress and impairing their metabolism, and then activating various stress response pathways that I'm not going to talk about today, but that I did look at in this previous work. This iron response pathway is also going to function, especially to work via this one response factor called DAF16, which I did find was upregulated to compensate for these different stresses. So with this being, or oxidative stress being an important part of this, I started to ask the question of whether or not I could perhaps reduce oxidative stress to rescue any of these phenotypes. And I mentioned early on that one of the phenotypes is that these mutants are short-lived. And I chose to use that phenotype and do a lifespan assay where we plate these worms in their very first stage of life called L1. And then this is just a Kaplan-Meier survival curve, and you check them every day, see if the worms are dead or not. And I either treated them with an antioxidant called N-acetylcysteine or a vehicle control. And you can see that that vehicle or N-acetylcysteine has no effect at all on wild type worms. But when I look at the two mutants here, I'll actually go to the Gly166 cis mutant first. This is very short-lived, and the N-acetylcysteine had no effect. So clearly it is insufficient to rescue that phenotype. But for the Gly147 mutant, you can see the rightward shift in the curve from the vehicle to the N-acetylcysteine, indicating that, yes, I was able to rescue the lifespan of that mutant by treating with an antioxidant, further supporting the idea that that oxidative stress is a significant contributing factor in those mutants. So to summarize what I've told you so far, I focused on these two mutants. And when I looked at respiration, it was impaired in the Gly166 cis mutants, but it was normal in the Gly147 mutants. When I looked at the mitochondrial membrane potential, this was the TMRE staining, however, it was impaired in both of the mutants. This started to make more sense once I realized that reactive oxygen species were predominantly increased in the Gly147 mutant, although there was that slight increase in Gly166 cis. And that antioxidants could rescue at least the lifespan of this Gly147 mutant, but not the Gly166 cis mutant. So clearly there were different mechanisms of dysfunction occurring between these two different mutants. So I'll come back to the questions that I'd asked, and I think that this has largely addressed just the first question. What's going on in the mitochondria? It is that NFU1 is having impairment in its dimerization around iron sulfur clusters. This leads to direct effects as well as some of the downstream indirect effects. And do any non-metabolic factors contribute? That's the data that I didn't show about iron, that indeed we do have iron accumulating, especially in the Gly166 cis mutant, and that is going to also result in mitochondrial dysfunction. But what about the impact that this has on the physiology of these organisms? So I mentioned early on when I was first talking about MMDS1 that it is a neurodegenerative disease, and one of the most common symptoms amongst individuals with MMDS1 is that they have neurodegeneration, and that dystonia, the loss of control of muscles, is one of the most common features there. More specifically, this presents as hypotonia, so loss of muscle tone, as well as spasticity, so a reduction in the actual voluntary control, and that these are amongst the earliest and most common phenotypes. And it turns out that worms are one of the best systems that we can use to study neuromuscular function, because they have muscles, they have motor neurons that operate basically the same as ours. And the easiest way to look at this is doing something called a thrashing assay, where if you put worms, so these are some worms you can see here, in liquid, they will characteristically thrash back and forth via this kind of sinusoidal motion. And we can do a lot of different analyses in these. And this data is in a paper that I published, I think, just last month. But we can look at a lot of the characteristics of that motion to try to parse out just what could be happening in their neuromuscular dysfunction. So I'm only going to focus on one feature here, and this is their wave initiation rate. How quickly do they generate thrashing motions? And it will consistently be shown as waves per minute. And what I found when I did this was actually not entirely what I expected. The Gly147Rh mutants had a faster wave initiation rate, whereas the Gly166 cis mutant, and indeed the NFU1 deletion as well, were significantly lower. They barely move when you put them in liquid. So given all of the other signs that the mitochondria were dysfunctional, I expected these two mutants to present this way, but I did not expect this hyperactivity in the Gly147Rh mutant. And it was really quite confusing, but this again shows that these different mutants have different effects. So I started to dive much deeper into this and started to think about what is happening at the neuromuscular junction. And I want to make a point here too, that this work was largely led by a post-baccalaureate fellow that worked with me. Her name is Pippa Rogers, and I will give her a shout out at the end of the slides too. But the work that I'm about to show you was performed by Pippa. So in both humans and worms, the way that our neuromuscular junction works is we have a motor neuron that is going to synapse onto a muscle. In humans, it's skeletal muscle. In worms, it's called the body wall muscle. The motor neuron stimulates contraction of the muscle by secreting the neurotransmitter acetylcholine. That acetylcholine will bind to what are called nicotinic acetylcholine receptors, which will allow sodium to enter the cell, the muscle, and to depolarize that and generate contraction. And we can look at the functionality of this system by using different agonists and antagonists of the neuromuscular junction. The one that I'm going to tell you about is a drug called levamisole, which is an agonist of those nicotinic acetylcholine receptors. So what we will do in the assay that I'm about to show you is you expose worms to levamisole, and in time it will cause them to paralyze because their muscles become hypercontracted. So that's what this assay here is that on the x-axis we have the minute since plating onto levamisole containing plates, and we have the percent of worms that are still moving on the y-axis. In black, we have the wild type animals, and then the same color scheme for the different mutants, including the NFU1 deletion here. And what I hope you can appreciate is that both of these mutants, and especially the NFU1 deletion, are shifted to the left, indicating that they are hypersensitive to levamisole. And this was actually not what I expected to see. Generally, a lot of the time that there is neuromuscular dysfunction, you have a reduction in the sensitivity to levamisole. So this was surprising but exciting. And it got us thinking about what could this reason be for the hypersensitivity to levamisole, meaning the hypersensitivity at the neuromuscular junction. And we thought, well, maybe it starts with the neurons. Maybe it's excessive acetylcholine secretion. Or maybe it's on the muscle cell. It's a hypersensitivity to the acetylcholine. Or maybe there's a reduction in an inhibitory signal. So I've only shown you acetylcholine, but there's also inhibitory neurotransmitters that can function there. If you lose those, that could also cause this hypersensitivity. Or because biology is not actually something that is normally binary, maybe it's a combination of all of the above. in fact, it is all of the above, but I'm going to focus on some of the data for the first 2 points here. So we first opted to look at whether or not acetylcholine secretion could be increased. And if we're again looking at this motor neuron here, we chose to look at this gene that in worms is called UNC17. UNC is short for uncoordinated, and we used RNA interference to knock down UNC17 because its normal function is to package acetylcholine into secretory vesicles. So if you knock it down, you get less acetylcholine into the vesicles, and that's going to reduce secretion. When Pippa performed the levamisole assay, you can see that we have in the same color scheme, but in solid lines, the empty vector, and in the dashed lines, we have UNC17 RNAi, so we have reduced acetylcholine. Surprisingly, this actually had, or not surprisingly, excuse me, a slight decrease in the sensitivity to levamisole in the wild type animals, but you can see that there's no difference at all in the NFU1 deletion. Likewise, there was no difference at all in the Gly166 cis mutant, but in the Gly147 mutant, this did nearly completely rescue the hypersensitivity that we had seen, indicating that there must be some effect of excessive acetylcholine signaling in that mutant. So we then performed the thrashing assay again using this RNAi, and you can see that as far as thrashing goes, the presence of UNC17 RNAi has no effect, but in the Gly147 mutant, we were indeed able to rescue that increase in thrashing, strongly suggesting that there is an excessive secretion of acetylcholine secretion in those mutants. This is particularly interesting if we think back to the fact that this is a rare disease model, because one of those 38 MMDS1 patients is homozygous for the Gly189 ARG mutation, which is the ortholog of the Gly147 ARG mutant. And this is the only patient to present with hypertonia, meaning that their muscles are hypercontracted. So this phenotype really made a lot of sense, given the fact that there could be this excessive secretion of acetylcholine from the motor neurons, and that is what's causing the increase in thrashing normally in this broader neuromuscular dysfunction. But we wanted to then continue going deeper into this question because it still did not clearly answer for us, is this exclusively a motor neuron phenotype or is it something to do with the muscle? So we opted to use CRISPR again and make tissue-specific knockouts of NFU1. So this is not working with the patient alleles at the moment. This is just a knockout in different tissues. We used a flip-frit system that was engineered by Peter Askier. And the way that this works is you flank the endogenous NFU1 sequence with FRT sites. This is short for flip-base recognition target. And then using a tissue-specific promoter, you express a flip-base, which in the cells where this is expressed, you cut out anything in between those two FRT sites. So because of the way we needed to engineer this, there was still the first exon of NFU1, but everything else is gone. So I'm going to first show you the thrashing assay that we performed with this, where we have, these are the control strains, which just have that tissue-specific promoter, but they don't have the FRT sites versus the NFU1 deletions. And in the muscle, we saw that there was a significant reduction in thrashing. And when we knock out NFU1 in the neurons, we see that there's a significant increase in thrashing. So this tracks with the previous data in that most of the mutants, the NFU1 mutants, have reduction in thrashing. That is starting to clue us in that that is probably a muscle phenotype. But if you lose NFU1 in neurons, that could induce this increase in activity. So we followed this up with the levamisole assays as well, and I'll show you first the muscle-specific knockout. Again, we have our control in red, and we see this leftward shift in the muscle-specific knockout of NFU1. And this, again, correlates with the thrashing assay, as well as what we expected to see based off of the other mutants. Now, what was very surprising to us was that when we looked at the neuron-specific knockout, that there was actually no effect at all in the sensitivity to levamisole. But that makes a little bit of sense when we thought back to the fact that levamisole is specific to targeting the acetylcholine receptor, which is on the muscle cells. So even though we've knocked it out, maybe this is just a little bit more complicated than we thought at first blush, that it's not directly connecting the neuron dysfunction to the muscles. And to be perfectly honest, I still don't have a great explanation for why the thrashing assay shows a difference, but we don't see that in the levamisole assay. But this is a somewhat more complicated phenotype to understand. So we next wanted to look at the complementary experiment. If it seems that both muscles and neurons might need NFU1, at least to some extent, what if we add it back in a tissue-specific manner in the patient-specific mutants? So we use something that is engineered called the ski lodge system for tissue-specific re-expression of NFU1. And this is a system that uses transgenesis, and you can add back a sequence using tissue-specific promoters. So I used either the worm NFU1 sequence, and I used the entire genetic locus because it's small enough that I could do that, and I didn't want to risk the chance that there was any sort of intronic regulatory sequences that I might be missing. So for the worm sequence, I'd use the entire locus, or for the human cDNA, and I'm not actually going to show the data for this right now, but I just had to use the cDNA because the gene is much larger in humans than it is in worms, but still, we wanted to test both of these. So when you combine this, or you insert these sequences into the transgene under the tissue-specific promoter, we could again look at either muscle-specific or neuron-specific expression or re-expression of NFU1. And I'm first going to show the data that we got from the thrashing assay with the Gly147R mutant. So this is similar to the data that I've shown before. I apologize, this should be black. It was just a clerical error on my end. But yes, we have this significant increase in the rate or the wave initiation rate of these mutants. Now, when we add NFU1 back to the muscles, it completely rescues that hyperactivity. And that was a surprising phenotype. Everything prior to this had been pointing to this mutant being more specifically affected, or more specifically affecting the neurons. But clearly, if we add back NFU1 in the muscles of these mutants, we can rescue this motility phenotype. Whereas when we add it back in the neurons, there is a very slight rescue, but not nearly to the effect of muscles. So this actually, I was hoping it would be this slam dunk experiment to really explain which of these mutants was more specifically important in different tissues. And it didn't give us that level of resolution, other than the fact that, yeah, this gene is important in both neurons and muscles. This Gly147R mutant in particular seems to have a diversity of effects. When I looked at the Gly166 cis mutant, and before anybody notices or asks, yes, this is the same wild type data that is being reused in these different figures. We've seen it is consistently significantly reduced in its wave initiation rate. But when I add NFU1 back to the muscles, it completely rescued their defect in that wave initiation rate. So for this mutant, again, the muscle seems to be more sensitive to NFU1 dysfunction than neurons. But when I did re-express this in neurons, you can see that, again, there was a partial rescue, although not complete. So this had led to my ability to make some conclusions in that the muscle must be more sensitive to these patient-specific mutations than the neurons, the motor neurons specifically. But we can't always draw the direct comparison between the tissue-specific knockouts and the re-expression because, again, we have the complication of the existing mutant alleles here. So if I summarize what I've shown in the second part of this talk, again, we have our two different mutants here. If we just looked at thrashing, the Gly147Rh mutant had an increase, the Gly147 or 166 cis mutant had a decrease, yet both of them were hypersensitive to levamisole, indicating that there was this difference or this impact on cholinergic signaling. Because of the fact that the UNC17 RNAi could rescue the levamisole sensitivity, I also looked at thrashing in that, where it was capable of rescuing both of those phenotypes by knocking down acetylcholine secretion. If we add into this figure the tissue-specific knockouts, remind you that thrashing was increased when we knocked out NFU1 in neurons, and it was decreased when we knocked it out in muscles. And that for the levamisole sensitivity, that the muscle-specific knockout was hypersensitive, whereas the neuron-specific knockout had no effect. So this was again starting to kind of indicate that it seemed like that NFU1 might be more essential in the muscles than in the neurons, and that this model was more similar to the Gly166 cis mutant, whereas this model was more similar to the Gly147Rh mutant, with lots of caveats in that. Then if we look at the re-expression, we can see that when we re-express NFU1 in neurons of these different mutants, there was slight incomplete rescue in both cases, whereas when we re-expressed NFU1 in both of those mutants, it more completely rescued their thrashing phenotypes in both of the mutants as well. So it really does seem that the muscle is what is particularly sensitive to dysfunction of NFU1, either whether it is completely absent or whether it is something that is more along the lines of a hypomorph. And so the way that I've been thinking about this and the way that I've been modeling it is, if we have our motor neuron up here and we have our muscle down here, and we have a mitochondrion in each, we've got NFU1 with its iron sulfur clusters that, by mechanisms that are not entirely clear to me at the moment, may impact the packaging of acetylcholine into cholinergic vesicles, secretion of those vesicles, and then the signaling via those nicotinic acetylcholine receptors, which are going to affect contraction. Likewise, the mitochondria in the muscles themselves are obviously also going to affect the ability of those muscles to contract. In the Gly147Rh mutant, which I will remind you does not bind as well, and it does have oxidative stress phenotypes, the results are consistent with there being an excessive production of acetylcholine. So that would also be an excessive secretion, although I haven't empirically shown that. The data is consistent with it. That is going to lead to contraction or hypercontraction in those muscles, giving them the hyperactivity, as well as it seems that the muscle-specific rescues also show that the muscle are contributing in this mutant as well. In the Gly166 cis mutant, it's not as clear that the neurons are as significantly affected, although that rescue construct did, or the re-expression construct did have some effect on acetylcholine secretion itself. It seemed to be more so coming from the muscle. And it's not really clear exactly at this point, like, is this actually contraction? Is there an elevation in contraction that is causing them to have this reduced motility, or is it just that they are incapable of responding to these signals? And exactly how this is happening is not clear, although I do have a couple of hypotheses. So to bring this back to how I started this talk of thinking about mitochondrial diseases, know that what I've focused on with a couple of specific alleles in MMDS1 is going to be causing oxidative stress in some of the mutants and not others, as well as acetylcholine dysregulation. And that we can parse this now into genotype-phenotype relationships between the different alleles, that ISC, or iron sulfur cluster mishandling, is really what is dictating this, and that maybe, just maybe, genetic diagnoses, if you can identify which allele an individual has, that this could impact therapeutic options for those patients, especially as far as acetylcholine is concerned. So with that, I would like to thank some people. The folks that are currently in my lab, so this is Kenyon College here, it's nestled into the hills of Ohio. None of these individuals actually worked on this project, but I do certainly want to thank them because they were brave enough to take on a brand new PI. But I really want to thank my postdoc lab. So Andy Golden was my PI who was fantastic in helping me get started in this project and then also giving me the freedom to run with it. I also really want to thank Pippa Rogers in particular, who did a lot of that levamisole work, as well as Sydney Kelly, who did more with the tissue-specific re-expression construct. So this is Pippa here, and here's Sydney. These are my collaborators at the NIH, as well as at Vanderbilt University. And since I am still fresh out of my postdoc, I do want to thank the NIH worm community who was really supportive. So with that, I thank you for your time and attention, and I'm happy to take questions.
Thank you so much for sharing your work or your team's work, Peter.
Yes.
Great, fantastic research going on there. It really is interesting. I'm sure we'll have lots of questions, so I'll move straight on to the Q&A now. Okay, so your first question is, have you tried performing chemical rescue on these phenotypes?
Yeah, so I showed the N-acetylcysteine rescue of lifespan, and it was able to rescue the Gly147Rh mutant, which really was part of what helped inform my idea that oxidative stress was in effect here. I did actually also test exposing both, or especially the Gly147Rh mutant to N-acetylcysteine again, and then looking at the thrashing assay and the sensitivity to levamisole. And surprisingly, there was no effect at all. So it would seem that it's not necessarily the reactive oxygen species or the oxidative stress that is causing these motility phenotypes. It must be something else that's going on in that mutant.
That's fantastic. Thank you. We've got time for a couple of more questions. So your second question is, this work focuses on acetylcholine for the excitation of muscle contraction, but is it possible that there is also dysfunction in the relaxation of muscle?
Yeah, so I mentioned when I first started talking about the neuromuscular junction that there's both the stimulatory or the excitatory pathway, which is acetylcholine, and then there are a few different pathways for relaxation or inhibition, the most prominent of which is GABA signaling. And I didn't share that data, but it is in my most recent publication that we also used the converse experiment rather than levamisole with a drug called piperazine that is an agonist of that GABA receptor. And we did see that there was an impairment in the sensitivity to GABA signaling as well. So we cannot yet rule out that there's also some sort of impairment in GABA signaling and in the relaxation of those animals. I will say, though, that I did a very, very simplistic test that we can do in worms where if you just touch them on the nose very gently, they should recoil away from that, and that is also controlled by GABA. That response was completely absent in the NFU1 mutant. So it does suggest that there's some impact here of just what's happening with GABA signaling.
That's great, thank you. I think we've got time for a couple of more questions still. And so your next question is, are these phenotypes restricted to the neuromuscular system, or are there other neurophenotypes?
Yeah, so clearly I focused on the neuromuscular system for my work thus far and for this presentation. And part of that being that the individuals with MMDS1 present with these neuromuscular phenotypes and that the worms are really effective for looking at that. However, I have done a little bit of preliminary work also showing that sensory neurons and those that control behavior are also dysfunctional. So I have a student who's working with me currently, so this is Kavya, who is looking into the chemosensory aspects of, or the chemosensory phenotypes of these NFU1 mutants. And this is a phenotype that's really well established or really well characterized in worms. And that means that we will be able to use different chemicals and figure out specifically which neurons are or are not functional in these mutants, and then try to determine why those specific neurons may or may not be sensitive to NFU1 dysfunction. So our hope is that by expanding this beyond just motor neurons, that we can get a broader idea of why the neurodegeneration that occurs in MMDS1 patients may be occurring.
Fantastic, Peter. I think we've got time for one more question, and that is, you modeled these variations as homozygotes, but many of your patients are compound heterozygous. Do you have plans for analyzing compound heterozygotes?
Yes, so this is a question that I get all the time. And the reason that I chose to analyze these specific mutants as homozygotes is because of the fact that most of the individuals with MMDS1 are compound hets, that the pathogenicity of the individual alleles is really unclear. And clearly my work has shown that these different mutations can have very different effects. As far as modeling the compound heterozygotes, this is something that I look forward to doing. I have not started doing yet, but it would be fascinating to see if, say, the Gly147Rh mutant crossed with the Gly166 cis mutant, if that somehow balances out in some way. I don't actually expect it would, because we still have NFU1 dysfunction, but it is an avenue of research that I look forward to pursuing.
That's great. Thank you so much, Peter. And unfortunately, that's all we have time for today. But any questions that we didn't have time to get to will be sent to you after the event. Okay. And as with all the other talks in the event, this talk will now be made to view whenever you want for the next 30 days. So that just leaves me to say thanks again, Peter, and we will be back soon with the next talk.
Thank you.